Hello my dear students, welcome back to another exciting journey through the wonders of animal biology. Today we dive into the fascinating world of tunicates, focusing on a unique phenomenon that is retrogressive metamorphosis in Ascidia. Unlike most animals, the free-swimming larva of Ascidia resembles a chordate, but the adult loses many advanced features like the notochord and tail. The transformation is known as retrogressive metamorphosis when the adult becomes simpler than the larva. So let's explore how this unusual life cycle challenges our idea of development and evolution in chordate. What the definition of metamorphosis? First of all, we study metamorphosis is a biological process involving a series of changes that a larva undergoes to become an adult. Now, what is retrogressive metamorphosis? In retrogressive metamorphosis, the larva exhibits advanced features which are lost during development. The resulting adult form is degenerated, primitive, or sedentary. Occurrence in animal kingdom. Retrogressive metamorphosis is rare compared to progressive metamorphosis. It is found in life cycle of Saculina, a parasitic barnacle on crabs, Urochordates, subphylum of chordates. This is the diagram of retrogressive metamorphosis in Saculina, and this is the diagram of retrogressive metamorphosis in Urochordates. What happens during metamorphosis in Urochordates? We have found that the chordate features are either lost or reduced. The larva transforms into a sessile adult, lacking many advanced features. Why metamorphosis is called retrogressive in Urochordates? The active free-swimming larva transforms into a sedentary simplified adult in case of retrogressive metamorphosis in adults. So the term retrogressive is used because development involves loss of complexity. So due to this transformation from an advanced larval stage to a simpler adult stage, the process is termed as retrogressive metamorphosis. So the free swimming larva will transform into a sedentary adult form. To understand retrogressive metamorphosis, let's first look at the early developmental stages. What is the pre-larval stages? During this stage, fertilization is external, producing transparent colorless all-acetal eggs, eggs with little or no yolk. Cleavage and embryo formation. Development starts with holoblastic and unequal cleavage. Entire egg divides unevenly. This leads to the formation of a coeloblastula. Gastrulation occurs by invagination, forming a three-layered gastrula. Larval shaping. The embryo gradually elongates and develops into a tailed Ascidian tadpole larva with an oval-shaped body and a laterally compressed tail. Larval stage, that is tadpole larva. The larval stage is known as the tadpole larva. It is highly active, free swimming, and feeds from the environment. Eventually it attaches to a hard surface using three adhesive papillae, tail facing upward. This marks the beginning of metamorphosis, during which the larva transforms into a sessile or non-motile adult. This is the larva of Ascidia and this is the adult form. Structure of a tadpole larva in Ascidia. This is the diagram of free swimming tadpole larva of Ascidia, which is a remarkable free swimming form that closely resembles the chordate body plan. It shows clear features that reflects its evolutionary position under the Urochordata. Size and shape. The larva is elongated and cylindrical, measuring approximately 4.2 mm in length and 3.3 mm in width. Body region. It consists of a distinct oval-shaped trunk or head and a laterally compressed tail. The tail bears a transparent caudal fin that runs along both the dorsal and ventral margin. Now, what is the protective covering? The entire body of Ascidian larva is enclosed in a protective tunic made of tunicin protein. Attachment organs. The head bears adhesive papilla or chin warts made up of cuticle which help the larva attach to a solid surface during metamorphosis. Tail features. The tail contains muscle bands aiding in active swimming. The caudal fin is remarked with the fine striations or striae considered precursors of fin rays found in fishes. Notochord. A rod-like notochord extends through the tail, one of the key chordate features. Nervous system. A dorsal tubular nerve cord runs above and parallel to the notochord. Anteriorly it enlarges into a cerebral vesicle that contains an ocellus, the light sensitive eye spot, and an otolith, which is a balancing organ. Neurosensory connection. The sensory vesicle opens into the pharynx through a neuropore. Yolk cells, a mass of pyramid-shaped yolk-filled cells, is located between the ectoderm and the sensory vesicle, providing nutrition. Mesenchyme, subectodermal mesenchyme cells are spread throughout the body, contributing to internal structure. Feeding and digestion, a mouth is present but covered by the taste or protective covering. As the larva matures, the alimentary canal degenerates since feeding stops post-attachment. Pharynx and gill slits. The sac-like pharynx is performed by two pairs of gill slits, that is stigmata, aiding in filter feeding. Endostyle. A well-developed endostyle is present which is homologous to the thyroid gland in vertebrates. Circulatory system. A non-functional heart is present ventral to the pharynx along with paired atrial sac and a pericardium. Behavioral traits. The larva is positively phototactic, moves toward light, and negatively geotactic, that is moves away from gravity, helping it swim freely in the water column before settlement. Metamorphic changes and emergence of the adult. The tadpole larva, after being a short period of free swimming life, becomes sluggish, fixes itself to the sea bottom by adhesive papillae, and undergoes first larval tissue degeneration. Tail reabsorption. What happens? Male tail muscles, notochord and nerve cord are gradually reabsorbed via phagocytosis. Body transformation. Ventral region grows between the mouth and the adhesive papilla. Tunic become thickens and forms a broad foot for permanent attachment. Pharynx and atrial cavity. The pharynx and atrial cavity become enlarged, filling most of the body. Stigmata, gill slits develop into pharynx. Nervous system degenerates. Brain reduces to nerve ganglia, ocelli, otocyst, and otolith disappear. Neural vesicle and axial complex are lost. Other degenerations, adhesive papilla, yolk cells, and mesenchyme disappear. Mouth shifts position, trunk broadens. Reproductive maturity. Gonads, either ovary or testes, develop and open into the atrium. Behavioral shift. Larva switches from positive phototaxis and negative geotaxis to negative phototaxis and positive geotaxis. This is the diagram of the Ascidian retrogressive metamorphosis from the larva to the adult. Progressive changes also occur during metamorphosis in Ascidia. The first feature is the pharynx enlargement. The pharynx becomes larger with more gill slits and the pharyngeal opening transforms into a ciliated funnel to help in filter feeding of the animal. Development of various organs. Liver and circulatory system begin to develop. A new structure called the velum is formed at this stage. Formation of reproductive organs. Gonad and their ducts develop from the larval mesodermal cells. Atrium expansion. The atrial cavity becomes larger and more spacious. Uneven body growth. Rapid growth occurs between the adhesive papilla or chin wards and the mouth, while the opposite side grows very little. Body rotation. Due to this uneven growth, the body rotates and the mouth gradually shifts to the outer or upper dorsal side. Now we'll study the loss of chordate features occur during larval metamorphosis in Ascidia. The first character is the complete loss of notochord and tail. Second one is the complete loss of sensory or cerebral vesicle, sense organ, and tubular nerve cord in case of adults. Nervous system is represented by a small solid nerve ganglion and a few slender nerves. All traces of metabolism are lost. The body undergoes rotation, thereby losing its bilateral symmetry. Thank you for watching this video. I hope this class on retrogressive metamorphosis in Ascidia helped you understand how an active free swimming larva transforms into a sedentary adult through remarkable process like phagocytosis, apoptosis, and differential tissue growth. This unique transformation not only supports dispersal but also offers key evolutionary insight into the origin of chordates from non-chordates, a truly fascinating journey. If you found this helpful, don't forget to like, share, and subscribe. See you in the next class. Bye.